Hello, my name is Lacey, and we'll be talking about CRISPR-Cas9, world-changing tool with my group members Chaylin and Chuxian. Before diving into the main topic, we need to establish what genome is. Genome is a term to describe all the DNA in our bodies. It determines your height, skin color, hair color, and much more. Most importantly, it relates to disease too. What is CRISPR-Cas9? Well, CRISPR-Cas9 is a gene editing tool in simple terms. Why is it so groundbreaking? CRISPR-Cas9 has many uses, such as curing disease, sustaining green energy, and so much more. CRISPR-Cas9 is divided into two parts, Cas9 and CRISPR. Cas9 is a bacterial RNA-guided protein that plays an immunological defensive role. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, is a group of DNA sequences found in bacteria and archaea. It is a system that forms a basis for the genome editing technology. Two things we need to know about CRISPR-Cas9 is that it is essentially made out of repeated sequences of nucleotides and spacers. Nucleotides are building blocks of DNA and are present throughout the gene code. Spacers taken from viruses that previously attacked serve as a memory bank. They recognize any foreign entities and fight them. Moving on, Chuxian will be detailing the mechanisms.
CRISPR-Cas9 system was originally found in bacteria as a defense system against viruses. That's right, bacteria has their own viruses, macrophage. This macrophage will transfer their DNA into the bacterial cell. When sensing alien DNA, the bacteria begins to activate the CRISPR-Cas9 system. The procedure goes like this. The bacteria produces two single-stranded RNA, one called tracer RNA, the other guide RNA. These two RNA are paired at one end, but the rest of the RNA is free and ready for binding. Guide RNA is one of the two most important components of CRISPR-Cas9. The other important component is an endonuclease protein called Cas9. You can see endonuclease as an RNA DNA scissors, which cuts a piece of DNA incorporated in it. The single-stranded RNAs settle in the Cas9 protein, forming a protein-RNA complex. The tracer RNA is a binding scaffold that keeps the complex together. The guide RNA is complementary to the DNA of macrophages, so it is a binding template for the macrophage DNA. The macrophage DNA unwinds and binds to the guide RNA, which also means the Cas9 cannot act upon the DNA strand. Like scissors, Cas9 clips off a segment of macrophage DNA, leaving the guide RNA intact, ready to bind to the next macrophage DNA. The damaged macrophage DNA is released into the cytoplasm, deactivated. How do scientists utilize CRISPR-Cas9? The system stays the same, while the guide RNA is different. Basing on the target DNA, scientists can edit the guide RNA to have corresponding base sequence. Then they place target DNA with CRISPR-Cas9 to have the desired segment clipped. With CRISPR-Cas9, scientists can edit genome easily in various ways. One way is to disrupt the DNA, rendering the whole strand inactive as the DNA cannot express itself. The second way is to remove a segment of the DNA. A segment of the DNA is removed, and the resulting two ends anneal together, producing a functional DNA with a few nucleotides removed. The third way is to replace the deleted segment with a desired segment, so the DNA can have different functions. Next is Chaylin on practical applications of CRISPR-Cas9.
Hi guys, my name is Chaylin and I'm going to be talking about how CRISPR is being used today as a tool for genetic engineering. So one example that is really pertinent to the field of green energy today is in the production of biofuel from algae. Algae naturally produce lipids which can be used as a greener alternative to fossil fuels. But one problem is that algae don't produce enough oil to make the extraction process profitable. Growing algae is also really water intensive, so if you have low yields, it discounts some of the green effects of using algae in the first place. So how do you solve this problem? Well, ExxonMobil, which is a big oil corporation, teamed up with Synthetic Genomics, which is a biotechnology company in La Jolla, to use CRISPR to optimize the algal genomes for lipid production. And by using CRISPR-Cas, they were able to boost lipid yield from 20% to 40 to 55%. In order to increase biofuel production, scientists first selected an algal strain native to San Diego that already had high levels of lipid production. Next, they used sequencing to identify genes involved in oil production. From this process, they identified 18 different genes that, when turned on, inhibit oil synthesis. So this is where CRISPR comes into play. CRISPR was used to bind to these sites and cut these genes in order to induce mutations that would inhibit their protein products. And by inhibiting oil inhibitors, you increase the overall oil output. Outside of the field of green energy, CRISPR is also being investigated to treat different genetic disorders. One example is promoting the production of fetal red blood cells in sickle cell anemia patients. And another is inducing mutations to T cells to increase tumor recognition in leukemia patients. Right now, CRISPR is also being studied as a faster and more accurate diagnostic tool in COVID detection. Thanks for listening, and I hope you guys have a great day.